Good afternoon. I am Nerea, and I'm here to present the results of our latest project titled Identification of a Memory-like NK cell population with enhanced ADCC activity in HIV elite controllers. More than 40 million people live with HIV infection nowadays. This infection targets mainly activated CD4 T cells, and if untreated, it leads to a progressive CD4 T cell loss together with immunological abnormalities. Antiretroviral therapy is effective at suppressing HIV viremia, but it is unable to cure the disease, mainly due to the formation of a latent HIV reservoir. However, we know that there's a small fraction of the population of people living with HIV that present spontaneous viral and immunological control over the infection in the absence of ART, and they have been designated as models of an HIV functional cure. This phenotype, the elite controller phenotype, is rare since less than 1% of the total population of people living with HIV presents it. And it is highly heterogeneous since the level of residual viremia, the time to reach the elite controller status, the level of immunological control, and the duration of this control can differ among individuals. On the other hand, we know that during HIV infection, a population of memory-like NK cells expand, and this expansion has been associated with protective effects in people living with HIV. These cells have been described to exhibit strong ADCC responses. However, in the context of persistent HIV control, the role of the phenotype and function of these cells remains to be elucidated. In our work, we wanted to study if NK cells from elite controllers exhibit memory-like phenotypic attributes. In addition, we wanted to know what was the functional profile of these cells and if it was different compared to other groups of people living with HIV. And finally, we wanted also to study the ADCC activity of these cells and if they presented this activity, if the response was associated to certain memory-like NK cell subsets. To do that, we used PBMCs from 5 different study groups: healthy donors, elite controllers that were divided in 2 different study groups. Both of them presented undetectable viral loads from 5 to 20 years, but the durable control group exhibited stable CD4 T cell counts during the patient's follow-up period. By contrast, the aborted control exhibited a progressive and significant CD4 T cell depletion during time. And we also included ART-suppressed and viremic patients. We performed phenotypic studies in order to assess the expression of important receptors for NK cell activity. And we analyzed this by multicolor flow cytometry, cluster identification, and reduction of dimensionality analysis. We also performed functional assays with isolated NK cells and 2 different cell lines and assessed the activation of these NK cells. And finally, we applied the results obtained from the phenotypic and the functional studies to isolate certain NK cell populations and perform the functional assays. First, what we did was to study the expression of different activating and inhibitory markers that are the ones depicted in the image by flow cytometry. And we wanted to consider the expression of all the markers simultaneously. So that's why we performed the cluster identification and reduction of dimensionality analysis. We were able to obtain 11 different NK cell populations based on the expression of the selected markers. And among these populations, clusters 2 and 4, that are the ones in green and black, were expanded in the durable control group compared to the ART and VHIR groups. These clusters presented high expression of CD16, an activating receptor that mediates ADCC, together with the chemokine receptor CXCR3, the inhibitory receptor NKG2A, and the memory-like NK cell receptors NKG2C and CD57. Next, we wanted to assess the functional response of these cells, so we performed co-culture experiments with the MHC devoid K562 cell line in the presence of IL-15. We performed the same analysis, and we were able to identify 3 different clusters, cluster 8, 9, and 10, that exhibited a strong cytotoxic profile, since they expressed CD107A, interferon gamma, and CD69 expression. In addition, 2 of these clusters, cluster 8 and 9, also express the memory-like markers. If we looked at the frequency of these clusters, upon stimulation, we observed lower frequencies of clusters 8 to 10 in the durable control group compared to the healthy donor group, the aborted control group, and both the ART and viremic groups. We also measured the natural cytotoxic activity of these cells in the presence of the cell line alone. And what we saw was that the durable control group exhibited lower natural cytotoxic activity compared to healthy donors and ART, and that these responses were positively associated to clusters 8 and 10, that were the ones exhibiting a strong cytotoxic profile. Finally, we measured the ADCC activity of these cells by co-culturing them with a latently infected cell line, the ACH2, in the presence of plasma from a viremic patient. And we saw that the durable control group exhibited a marked ADCC response against HIV-infected cells compared to the rest of people living with HIV groups. In addition, this response was positively associated to specific NK cell populations, such as cluster 2, 4, and 1, that, as you saw, expressed the receptors CD16, some of them CXCR3, NKG2C, NKG2A, and CD57. To confirm these results, what we did was to isolate via FACS sorting populations expressing a different combination of these receptors. As you can see, population 3, that was the one expressing CD16, NKG2A, NKG2C, CD57, and CXCR3, was the one that presented enhanced capacity for killing HIV-infected cells via ADCC. So what we can conclude from this work is that there's a population that express CD16, NKG2C, CD57, NKG2A, and CXCR3 that is expanded in the durable control group. That NK cells from durable control individuals exhibit reduced cytotoxic responses upon stimulation, but robust ADCC responses against HIV-infected cells. And that the population that is expanded in durable control group presents ADCC, an enhanced capacity for ADCC-mediated killing of HIV-infected cells. We believe that this work provides important clues for the design of new NK-based immunotherapies to control HIV. And finally, I would like to thank all the people that participated in this study, especially my supervisor, Dr. Maria Buzón, and all the study participants.
Thanks, Nerea. So, questions for the talk?
Thank you very much. Really nice presentation. So I wanted to know, what do we need to do in order to have this type of NK cells in HIV-infected individuals?
We are conducting further experiments. We are doing single-cell RNA sequencing of these populations to see what do they express. But we are also trying to expand these populations expressing the double markers, and we are still setting up the experiments, so we don't know. But we know that the individuals that present this control exhibit these populations.
Thank you.
Do you have any information on the intracellular molecules on these cells? If they are specific also, you have like adapter molecules or?
We don't know that.
Okay.
Thanks.
I ask the same every year. So memory-like NK cells, the ones that express NKG2C, have been described to develop in response to CMV infection. And as far as I know, there is no any other situation, context in which these cells appear in the absence of CMV infection. So these subsets or the presence of these cells can be amplified by co-infections, for example, in HIV patients, but the origin is always due to CMV. So I was wondering whether, when you compare the different groups of individuals and the presence or absence of these cells, whether all those individuals are CMV positives. Have you taken that into account? For example, in the comparison with healthy donors or in the comparison with the other HIV groups?
That's a very interesting question, and we are looking at this information. We know that the R-treated cohort, we only have, from the total number of patients that we have included in the study, we only have the information of half of them.
The phenotypic characterization of these cells, so till now has been understood as a memory-like NK cell, adaptive NKG2C, and memory-like NK cell are cells that express NKG2C but lack NKG2A, which is the inhibitory counterpart. So it's curious that most of your enhanced activity, according to your analysis, is accounted by this weird NKG2C positive, NKG2A positive subset. So perhaps you want to go deeper on that, since it's perhaps the most novel thing.
Yes, it was very surprising, and the samples that we have sent to our single-cell RNA-seq are the population expressing the double NKG2A, NKG2C, and two expressing or NKG2A alone or NKG2C because we want to know more. But also in our group, we're trying to expand these cells, and we are trying to try the cytokine-induced way. And these cells do express NKG2A.
NKG2A is upregulated upon activation, particularly if you use high doses of IL-2 or IL-12. So just be careful with that, because this is just a transient upregulation that may disappear or not, and it doesn't mean that it's the counterpart of the NK cell that you find in vivo. So if you use a protocol of cytokine-induced memory-like NK cells or any protocol for NK cell expansion that uses feeder cells, you will have always NKG2A upregulation in 80% of the cells. So
yeah,
just take care of it.
Thank you.
Any other questions?
Hi. In your experiments, have you also tested in the ADCC, have you tested cytokine responses?
No.
Because in the memory-like cells induced by HCMV, they are particularly good at making cytokines, like more than cytotoxic effect, they are proficient in cytokines. And I was also wondering if you have checked the other markers of adaptive NK cells, like gamma chain negative cells, PLZF negative, which are also other markers of adaptive NK cells that can overlap with NKG2C. And also maybe this NKG2A that you see is a sign that they are kind of activated in vivo from the infection?
We haven't checked for the cytokines. We only checked for the cytotoxic activity of these cells, besides the phenotypic characterization. And what was the second question, sorry? You have checked for other markers of adaptive NK cells? No, we had limited flow cytometry panels, so we did not. Yeah, we don't know if there's maybe this phenotype and what occurs, it is a hypothesis, it's that maybe they do express more anti-apoptotic molecules, so they are in this state instead of more activity. We don't know. So we hope that with the results of the single cell, we know a bit more. Thanks.
Any more questions? Okay, thank you, Nerea.